I'm Darren Carell. In this video, we'll discuss, in a relatively detailed way, a specific pharmacodynamic principle, that being an opioid's mechanism of action on a cellular level. Recall, opioid receptors are a group of inhibitory receptors located throughout the body, specifically multiple areas of the brain and brainstem, the spinal cord, peripheral nervous system, joints, lungs, heart, immune and endocrine systems, and the biliary and GI tracts. Structurally, opioid receptors are 7 transmembrane G protein coupled receptors. That means that they cross through the cell membrane 7 times, and that they act through a G protein, a type of transducer, which detects molecules outside the cell, the opioid ligand, and on binding, the G protein becomes active. And then, through a number of next steps, like interacting with an enzyme or an ion channel, various internal pathways occur that cause a cellular response. Any receptor that acts through a signal transduction pathway like this is called a metabotropic receptor. This is in contrast to receptors that directly affect ion channels, aptly called inotropic. We'll use opioid receptors at a synapse in the nervous system as our example for the discussion of their mechanism of action. However, in order to understand how opioids work, we have to have a slightly more in-depth discussion of neurophysiology than what was covered in the important nomenclature presentation in the introductory series of videos, or the document of the same name on the website. So, the normal course of events at a synapse is that the action potential arrives at the end of the presynaptic nerve. Calcium channels open, calcium flows in, the enzyme adenyl cyclase is activated, increasing the amount of cyclic AMP in the cell, which results in neurotransmitter release. Note, this shows co-release of different neurotransmitters from the same vesicle. Depending on the type of nerve, the neurotransmitters released may be of just one type or more than one type, but each released from separate vesicles. But all this is really beyond what we need to worry about here. Now, in nociceptive pathways, the 2 most important neurotransmitters released are glutamate and substance P. More on them in other video series. Neurotransmitters bind to the receptors on the postsynaptic membrane. In nociceptive pathways, this is the AMPA and NK1 receptors, also discussed more elsewhere. AMPA activation by glutamate directly opens an ion channel and allows sodium to flow into the cell. Thus, it is an inotropic receptor. The NK1 receptor is G protein coupled, or metabotropic, and activation by substance P leads to many complex responses, including release of calcium ions from intracellular stores and inhibition of the normal outward movement of potassium. This overall increase in positive charges in the cell results in depolarization, and the action potential will travel along the nerve. Okay, now back to opioid receptors. At synapses in the nervous system, opioid receptors exist presynaptically and postsynaptically. All 4 types of receptors, mu, kappa, delta, and nociceptin, have similar intracellular responses following activation by an agonist. On the presynaptic side, when an opioid binds to its receptor, the G protein becomes active, and this proceeds to reduce calcium influx into the cell. In addition, the activated G protein also directly inhibits the activity of the enzyme adenyl cyclase. Both actions result in reducing the amount of cyclic AMP produced, which prevents the release of the neurotransmitters. So the nerve fibers can't communicate with each other and the nociceptive input is stopped. On the postsynaptic side, when an opioid binds to its receptor, the G protein becomes active and interacts with specific ion channels to enhance potassium efflux from the cell. This reduction of positive charge in the cell results in hyperpolarization, and thus the nerve cannot conduct an action potential and nociceptive input is stopped. This all likely seems quite complex, which it is, but in fact, these are only the mechanisms of action for certain of the effects of opioids, like analgesia. Some of the other important mechanisms will be discussed in other presentations. I hope you learned something from this talk. Please look for the other presentations in the opioid pharmacology series on the website. And as always, thanks for watching.